Cool. All right. Thanks a lot for giving me the opportunity to present. So I'm an assistant professor at Montana State University, and I want to talk about some new correlative microscopy approaches that my lab has been developing to get in collaboration with a couple of other labs. So the problem we're trying to solve is that outside laboratory-based experiments, microorganisms live as members of spatially structures and taxonomically distinct and metabolically diverse and interdependent communities, which are exposed to varying physical chemical conditions over time. And so the underlying problem is that typically how microbial ecology studies these days are done is that they are most often bulk analysis that don't do a good job in capturing this intrinsic heterogeneity of microbial communities. And so one of the things my lab is trying to do is to overcome this problem by developing new techniques that work at single cell resolution to study these communities at a little bit finer detail. So sorry for that. Let me just point this. So I want to talk about two studies my lab has been working on. The first one is one that was published, came online just half an hour ago. So I just quickly added this slide in environmental microbiology that was funded by the Moore Foundation's Marine Microbiology Initiative, in which my lab and postdoc Dr. Rachel Spitz, in collaboration with former postdoc of Pete Gergis's lab, Jeff Marlowe, who now runs his own lab for about a year at Boston University, and the lab of Mark Ellisman has been working on to try to study the in situ activity and interaction of microorganisms on a sub-micrometer to centimeter scale. And so the idea, the underlying technique that enabled this, I think, is an approach that my lab calls substrate analog probing. So this is the idea that you use synthetic compounds. So for example, synthetic nucleotides, synthetic amino acids, synthetic fatty acids, synthetic diamino acids, that if you add to a sample, incorporated into the respective biomolecules, DNA, RNA, proteins, fatty acids, and peptidoglycan, instead of the natural counterparts. And so with this, we can use click chemistry enabled fluorescence staining methodology to, under as close as possible in situ conditions, visualize newly synthesized microorganisms that synthesize new DNA or new peptidoglycan or new membrane lipids, or what we have been focusing in this study, new proteins. So the idea is that you add an amino acid analog that is incorporated instead of methionine into cells, pretty much by all microorganisms that we have ever tested pretty much, and that you can couple with other fluorescence microscopy techniques, like for example, fluorescence in situ hybridization, to then visualize in a complex microbial community who are the organisms by fish and which of them are making new proteins under a given set of environmental conditions. And so in this work, we have been combining this with some sophisticated electron microscopy and EDX analysis to try to visualize microbial activity directly where they live. And our study site for this was Little Sepowiset Salt Marsh, which I think many of you know. This is a very long microbial ecology site outside of Woods Hole in Massachusetts, where we took sediment cores, we brought them back into the lab, and then essentially replaced the water in this course with water from the same site, to which we simply added this synthetic amino acid called HBG. And then to some cores, we also added complex carbon sources to basically see how the microbial community responds to the addition of different carbon sources. We then put them back into the environment. So we left them in situ for about 4 days. And we simply sealed off these tubes using gas permeable but water impermeable membranes. We left them for 4 days. After that, we came back. We took the samples, we embedded them in an electron microscopy grade resin, we sectioned them and analyzed them using a combination of electron microscopy and energy dispersive X-ray spectroscopy. And we then overlaid the information that we got from fluorescence microscopy, electron microscopy, and EDX all together. In addition, as you see in this picture, we had parallel cores that were treated differently, in which the individual cores were sectioned in 1 centimeter increments, and in which the translationally active microorganisms that were fluorescently stained, right, were simply sorted using fluorescence activated cell sorting, and the taxonomy of these organisms was revealed using 6S RNA gene sequencing. Okay, so this gives you just an idea of how the data looks like. I apologize, I know this is a very dark slide. It looks better, believe me, than if you invert the colors. But so the idea is, right, so here we have, a particular field of view that is about 150 times 100 microns, how the sample looks in electron microscopy. In red, you see translationally active biomass. In green, you see cells that contain DNA but are not translationally active or have not taken up our amino acid. And here you see, the different elemental maps that we got from our EDX analysis, so calcium, sodium, and so on. And we can then overlay all this information into a big piece, a masterpiece, so to speak, in which we can then identify the mineral phase based on the elemental composition of the individual mineral and sediment particles. We have the biomass that is translationally active in red, and the green pieces, the green biomass is not translationally active, but DNA containing cells. And with this, we can then study whether or not biomass that is associated with different mineral particles are more or less translationally active, depending on where they are in the sediment pore space. So one other thing we can do with this, right, because we have mineralogical information, we can do some image analysis and then determine and say, okay, if we bin the biomass in, let's say, 1 micrometer, 5 micrometer increments, as how far away they are from a particular mineral particle or from other cells, we can then say, how active or how much of the biomass is anabolically active depending on how far away they are from a particular mineral. And because with mineral information, we can also do this, on particular mineral types. So in this particular case, we looked at 15 different quartz particles, and we saw that about 10–15% of the total biomass is within 5 micrometer away from these quartz particles and are also translationally active. An additional 12% or so are also within a distance of 5 micrometers, but are not translationally active. And we can play around with these different mineral phases to determine, how biomass is associated with it and is physiological active. And then we can bring this together with our separate data set, right, where we sorted cells based on their fluorescence, so based on their translational activity, and use 6ns RNA gene sequencing to determine in 1 centimeter increment into the sediment core, are particularly taxa of organisms more or less translationally active. So here is just an example. On the y-axis, you see in 1 centimeter increments, we go into the sediment, and we see a particular group of organisms called Desulfobacterales. This is a group of delta protobacterial sulfate reducers. And you see the yellow bar indicates they are more active, they get more active in particular sediment horizon than in others. In turn, we have other organisms that are less active deep into the sediment. So the blue bar indicates they have lower activity. They are enriched in the inactive fraction rather than the active fraction. And overall, we see that the trends that we would expect hold true. So we have some good sanity checks, for example, by this group Chromatiales. So this is a known group of photosynthetic organisms that are very active in the top 1 centimeter horizon, where there's typically sunlight reaching still in. And then the deeper you go into the sediment core, the less active they are. And so by the end of the day, we hope, right, to bring also in fish analysis into this, so that we can then overlay not only the translational activity in a particular sediment horizon and overlay this with the electron microscopy data, but also say, okay, let's look at individual cell resolution members within the group Desulfobacterales against we have a fish probe available. Another way we're trying to look at this that is complementary to what I just described, but somewhat independent, is using another lens at which we can look at anabolic activity, and that is stable isotope probing, specifically in combination with Raman microspectroscopy. So this is the idea that if you, feed organisms an isotopically labeled substrate and use Raman microspectroscopy to probe the chemical makeup of individual cells, so we get about 0.3 micron resolution here, you can see whether or not cells have taken up the heavy isotope by specific shifts in the Raman spectrum of the cell. And I show this here as an example. These are simply E. coli cells here that were grown in the presence of C13 labeled glucose as compared to C12 glucose. And you see that specific peaks that are indicated for the incorporation of C13 appear in the spectrum of the labeled cells versus the unlabeled cells. And so we've been combining this CYP Raman technology with these other electron microscopy and fluorescence microscopy approaches to come to another complementary approach of how we can visualize microbes at a single cell level. And so this was done in a collaborative effort by two fantastic graduate students of mine, Anthony Kotz and George Scheible, that basically came up with this surprisingly comparatively simple protocol where you take a microbial community, you apply your stable isotope label. In this case, we simply did deuterated water labeling as a general proxy for metabolic activity or anabolic activity, I should say. You then have some sample preparation steps where you basically just mark where your cells are that you're interested in. You then perform STM and EDX analysis. You do Raman spectroscopy. You do FISH, which identifies the organisms of interest. And if you want to, you can also go into the nanosims. And so this is a super correlative microscopy approach, right, in which we can overlay the biochemical information from Raman, the taxonomy from FISH, the elemental composition from nanosims EDX, and the morphology, or ideally even ultrastructure from electron microscopy, all on top of each other, all on the same biomass. And so we have been benchmarking this using a couple of different pure cultures, specifically here just a bacterium, E. coli, and the Archeon methanosacina that we just simply mix together. And then the individual cells were probed using Raman spectroscopy. You see here the CD peak, the carbon deuterium peak. So these are cells that picked up deuterium from the heavy water we added to the sample. And some of them are not active and some of them are active, which is consistent with the idea that we incubated E. coli in the presence or in the absence, as well as the Archeon in the presence or absence of deuterium. We then probe the same cells using FISH, so we can distinguish who is an Archeon, who is a bacterium. In theory, you can use any FISH probe you want to. Here, because we have a mock community of just two organisms, this was very straightforward. And we overlaid with the electron microscopy data. And in the end of the day, we can generate this, fake overlays basically, in which we combine all this information together, where we have the taxonomy, the structure of the cells, and the biochemical, the anabolic activity of these cells. And so we are currently trying to apply this to a bunch of different enrichment cultures and in situ samples we have in our lab. Ideally, we want to use this in a new project that was funded by the Moore Foundation and the Simons Foundation on new ASCAD archaea. So these archaea that show resemblance to eukaryotic cells, at least to a certain extent. And recently, we've been applying this to another very cool organismic group we are studying in our lab, these multicellular magnetotactic bacteria. So these organisms are uncultured delta protobacteria that are the only known example for obligate multicellularity within the bacteria, and also within the archaea, so within the quote-unquote prokaryotic realm. And so the way these organisms divide is that they don't have a single cell stage, but these consortia of cells that are arranged in these spherical shapes basically just simply grow in volume and then just split in half, basically, without having a single cell stage. So these are the only known example of obligate multicellularity. And so we have been combining these FISH, EM, Raman spectroscopy techniques to basically study these organisms that we have in the same site I showed you before, in Little Zipper with its Saltmarsh, to basically study whether or not different cells within the same consortia or different consortia are metabolically distinct or show different metabolic activity from each other. So with that, I'm just gonna stop here and I would be happy to take any questions. And this just shows you an overview what other things my lab is up to these days. Thank you.